evening, everybody. This is Julia Frost-Davies. I'm coming to you live from Mansfield, Massachusetts. If you can hear me, can someone give me a thumbs up or a wave so I know you're out there? All right. Thank you. Welcome to Tina's Wish, formerly titled The Boston Gathering, tonight titled Boston and Beyond. Welcome back to those of you who have attended with us live in Boston or other events across the country. And welcome to our new friends who have not before attended a Tina's Wish event. We are thrilled to have you with us. For those of you who have not been to one of our events before, Tina's Wish was founded in honor and at the direction of my former partner, mentor, and friend, the late Tina Brosman. She was an extraordinary woman, the former chief judge of the United States Bankruptcy Court for the Southern District of New York, a major presence in the restructuring field, and an even more phenomenal person. And when Tina was diagnosed with late-stage ovarian cancer, she asked us to devote all of our time, efforts, and resources to try to find an early screen so that other women would not suffer the same consequences and would have a fighting chance. When ovarian cancer is diagnosed at early stages, like stage 1, it is much more curable, treatable, and therefore survivable. The problem is, as you will hear from our medical professionals who are doing important research in this area, there is not yet an effective early screen. And that is why we are here tonight and why we are so thrilled to have your support. So before we dive in with our doctors, I'd like to have a short video for you to enjoy.
When I was diagnosed with ovarian cancer, the mission of Tina's Wish became very personal for me. I had a lot of doctors telling me nothing was wrong, and then I get this phone call, and it's like your whole world turns upside down.
There's absolutely a need for better early detection methods for ovarian cancer. If I have someone who comes in with stage 1 disease, I have a close to 90% cure rate. Unfortunately, if someone comes to me at stage 3 disease, my cure rate goes down to about 27%.
I did 6 rounds of chemo, and I got the NED, which is no evidence of disease. The key to treating this disease is early detection. And because of Tina's Wish, we're really close to getting there.
The woman you just saw in that video is an amazing person, Michelle Correal. She is an ovarian cancer survivor and a friend of Tina's Wish. And it is because of women like Michelle that we all continue this important work. I'd like to turn this over to my friends and colleagues and stalwart Tina's Wish supporters, Jim Wallack and Rebecca Matthews, who will introduce our medical speakers for the evening. Jim?
Thank you, Julia. Hello, everyone. My name is Jim Wallack. I'm a director at Goulston Storrs, and I'm proud to be one of the co-chairs for this evening.
Good evening, everyone. My name is Rebecca Matthews, and I'm also honored to be one of the co-hosts of Boston and Beyond tonight.
Rebecca and I have some very personal connections to Tina's Wish. My mother passed away at the age of 60, just 9 weeks after she was diagnosed with ovarian cancer. And Rebecca's sister, Rachel, is an ovarian cancer survivor who just marked her 4th anniversary of her last chemotherapy treatment. So we want to express our deep appreciation for the work of our terrific researchers and the generosity of our sponsors. Thanks to those sponsors, the evening has already raised $36,500. And we're really pleased to announce a match challenge for all of you. A group of 4 anonymous sponsors, donors, will together match the first $5,000 of additional funds raised during tonight's program. Please consider donating any amount comfortable for you during these very challenging times. Think of the women in your life. All of them deserve a fighting chance against ovarian cancer.
To donate, please visit tinaswish.org/donate. If you have questions for the doctors, please type them into the Q&A to the right of your screen, and we will take some questions following their presentation. Now, I am very excited to introduce Tina's Wish researchers, Dr. Dipanjan Chowdhury from Dana-Farber Cancer Institute and Dr. Kevin Elias from Brigham and Women's Hospital. Dr. Chowdhury and Dr. Elias's labs are working together, leveraging the power of artificial intelligence to develop a technique to detect ovarian cancer early and accurately. Before starting, I would also like to thank Dr. Kevin Elias for currently serving on the front lines of the COVID-19 ICU unit at Dana-Farber over the past couple of months. Thank you for your service and for saving lives. Now, a very warm welcome to Dr. Chowdhury and Dr. Elias.
Thank you, Jim. It's my pleasure to be here, and I want to first thank Tina's Wish for doing this in these troubled times, and I want to thank all of you here. I know zooming into this sounds cool, but after a day of being in front of a screen, it's probably not fun to be again in front of a screen. So I really appreciate the time and the effort. So I will try to take you through the early part of how we got to this stage, and I think Kevin is going to bring it home to tell you exactly what we are doing now. So the numbers for ovarian cancer, I mean, most of you may be aware of this, but it's actually quite disturbing. So one, there are about 250,000 women who get diagnosed worldwide and 140,000 deaths. 1 in every 75 women. So, people think of this as a rare disease. It's indeed rare, but not as rare maybe as we think. And I think the most striking number to me is that it is the most lethal gynecological malignancy. So if you look at the bottom right panel here, the percentage of women who get ovarian cancer and who don't make it is significantly higher than any other women's cancer. So this is something which at least I didn't know until I started working on this topic. And here is why we do what we do, early detection. So look at the numbers here. If serendipitously a patient gets to the clinic at stage 1, it really is luck here. The survival rate is over 90%. Basically, we can cure this disease. But unfortunately, 70% of patients get to the clinic at stage 3, stage 4, and the survival rate is below 30%. We are talking about a 60% increase in mortality. I don't think there is any drug that can do that with this sort of efficiency. It's really about detection, and that's really what got us motivated to work on this along with Tina's Wish. So the question I think we get asked very often is, why aren't there blood tests for ovarian cancer? There's a mammogram, there's colonoscopy, why not for ovarian cancer? So, well, what can I tell you? The only thing that is out there, which is a test, CA-125, which has been there since the 1980s, again, was discovered in the Longwood area here in Boston, HE4 in the early 2000s. These tests in blood are very useful in monitoring the disease. So when somebody has the disease, the doctors can really use these to monitor the disease. But they are really not good for early detection or for any screening program. So the bottom line is, right now, there is no test, there is no scan that can detect ovarian cancer early. That's something which everybody really needs to understand. There is no option, zero. And we really have to look beyond the traditional class of biomarkers. That's really what I want to get to. We really have to think outside the box. So here is the biology which drove us or got us here. About 20 odd years ago, the human genome was sequenced. And right about that time, as a community of biologists, we realized that only 2% of the genome makes proteins. What does that mean? So every gene that you have associated, that you have heard of associated with human disease, the BRCA gene, the BRCA gene, the RAS gene for lung cancer, the BRCA gene for ovarian and breast cancer, all of these just come from 2% of your entire genome. All the drugs that we are talking about, which are directed to specific targets, again, it's all directed to that 2%. So for 70 years, of molecular biology, it's all being directed to understand the 2%, and we have completely ignored the rest of the genome. And even about 15 years ago, the textbooks would read that the rest of the genome, the rest of all you see here, the red and blue, is junk DNA. Now, with advanced technology and with all the other tools that are coming up, we know that there is no such thing as junk DNA. 80% of the genome is making RNA, which is what we call non-coding, which means it's not making protein. I have to digress here with the COVID-19. I think everybody around now reads about RNA. So we know how powerful this is, a molecule that in this virus can kill 100,000 people in this country. Imagine our cells are full of RNA and we know nothing about what these RNA molecules do, or very little really. So my lab in the past 12 years has been working on one class of this non-coding RNA called microRNAs. These, exactly as the name sounds, are small bits of RNA. And these microRNAs have been shown to really influence the 2% of the genome that I said we know a lot about, and that we know is very important. So about a decade ago, it was discovered that microRNAs can be in blood. You could have microRNAs in blood like you have cholesterol or you have glucose. And as we started looking at it closely, we found that there were about 250 microRNA molecules, different, like an alphabet, like 250 letters in blood, in your circulation. And what we came up with, the idea is, can we capture these and sequence them and try to see patterns that show us that a person has cancer or not? And we actually did that and published this in a peer-reviewed article a few years ago, where we could say we can detect ovarian cancer looking at a pattern of microRNAs in blood. Let me walk you through exactly what we did. So basically, from a vial of blood, we can isolate the RNA, and then we can use a machine to actually measure this RNA with very high accuracy, the different amounts, changes in the RNA. And then comes this voodoo, the machine learning approach, which allows us to really know which pattern is showing the cancer. It's very much like now Netflix knows exactly which shows you want to watch. I know we are all watching a lot more Netflix than we ever did during these last 2 months. So the more you watch, the better they know what you want to watch next, or for the more intellectuals, the better documentaries you want to watch. So just to give you a better understanding of what this really is, one analogy we came up with is that when we do the sequencing of the blood, it looks like the night sky. Basically, all these stars are, imagine the different microRNA molecules. But as the pattern starts emerging, it looks like a constellation. It's the same pattern from any part of the world. Whether you draw blood in India or Poland or here in Boston, the same pattern that shows cancer starts to emerge. So that's basically what we developed in this paper. And what we could show is that at this early study, that we could detect ovarian cancer with about 92% accuracy, which is way more than anything out there with these microRNAs. And the other point I want to make here is that this is specific to ovarian cancer. So this particular pattern that we discovered was specific to ovarian cancer and not other cancers. Each cancer, we believe, would have its own pattern, and that's something we want to capture. But for now, we do believe we have a pattern which really captures ovarian cancer patients. So the question now is going early. And this is where I want to hand over to Kevin, who will take you to the next.
So the question we always get asked is, we talk about early diagnosis, and really how early is early? So one of the things we did immediately after that first publication was to focus our assay only on early stage patients. So only those patients with stage 1 or 2 disease, where the cure rates are the highest. And so we constructed a cohort of patients with 75 early stage patients, matched them with 100 perfectly healthy women and 100 women with benign tumors. And what we showed was that the microRNAs that we had published in our original signature were also the ones that could pick out patients with early stage disease from these patients without cancer. And what was particularly important was the specificity here was extremely high. So we talk about the negative predictive value, meaning if the test comes back saying someone doesn't have cancer, how much can we trust that test? And we're saying with 99.97% accuracy, so only 3 out of every 10,000 patients that we would say didn't have early stage cancer, in fact, would not have cancer. Now, the converse of that is that we would be able to pick up more than half of the patients who have had truly early stage disease. Now, what about moving even earlier? And one of the things that we looked at to say, well, we really want to diagnose women before they even present with that type of mass, is we looked at the tissue level. And we could find that in patients who had pre-invasive disease, so not even quite reaching the level of cancer yet, but in tissues that were destined to become cancer, if we looked microscopically at these early lesions, these early precancerous areas in the fallopian tubes that become most ovarian cancers, we could actually identify the microRNAs in these pre-invasive lesions, suggesting that what we were looking at was something that was inherent to the biology of early stage tumors, and that's carried through all the way to more advanced stage disease. But even in patients who don't actually have symptoms yet, we can start to see these microRNA changes. So where are we going now with this? Well, actively, we've been collaborating with the NIH, testing the ability of our microRNAs to predict the future diagnosis of cancer. Because we're not just interested in saying, right now, what's the likelihood that you have cancer? We want to know what the likelihood that someone's going to develop cancer in the next 1, 2, 3, even 5 years. And so because of our previous work, the NIH has been generous to give us access to the prostate, lung, colorectal cancer, ovarian screening trial, which was the largest study ever done to try to look at early diagnosis for a range of cancers, with ovarian being one of the largest parts. Now, this study, which occurred over 15 years and included thousands of women, we've been given access to about 200 patients who were diagnosed with early stage disease and been able to match them to controls from across the United States. And we've been able to pick subjects who had cancer diagnosed at stage 1 and 2, but had a blood sample within 5 years of a diagnosis. Not only that, we have several patients within the study with serial samples, so we can look to see how our signature changed over time as a diagnosis approached. So this is going to be our first large study in the general population in a clinical trial that we already know the outcomes for, to see how our early diagnosis signature would work in the real world. Now, we don't want to just be able to assess cancer. We want to really be able to help stratify people by their underlying risk. And what we've found is that using someone's underlying genetic risk, so for instance, whether or not they're a carrier of the BRCA genes, we can tweak our model to use a standard risk adjustment or a high-risk model. We know that in the general population, the chance that someone has ovarian cancer is quite low. It's 0.06%. Now, again, in that population, our specificity is extremely high. We generate very few false positives, on the order of 3 in every 10,000, but we're able to pick up more than half of the cancers. Now, conversely, when we apply our model to the high-risk population, where the chances that someone has cancer are about 1 in 100, we have very similar specificity, meaning very low false positive rates, but at the same time, we pick up virtually all the cancers in these women who are at the highest risk. So knowing that, we've launched a second study, which is called the MIGHTY study, or the microRNA detection study. And this is being run in collaboration with Judy Garber, who runs our genetics program at Dana-Farber. And we're taking women who are coming into us either through our GYN oncology clinics, where I sit, or the breast clinics, and have had a genetics referral. We know that they have, either based on their family history or their underlying genetic predisposition, an increased likelihood of developing cancer. And we're following these women prospectively. We're getting blood samples every 6 months until either they decide to have risk reduction surgery, or surgery for some other indication, or they're diagnosed with cancer. And this is going to give us our first real-world prospective analysis of how many of these cancers can we actually pick up before the patients would otherwise be diagnosed. And what we're hoping is, with the support of organizations like Tina's Wish, that we're going to be able to grow the Mighty cohort. So as of September, we've been enrolling patients in the Mighty trial, and we're hoping to be able to expand this study out where we can collect blood samples from around the United States, getting a more diverse population, accruing our target goal of 500 patients in a shorter time interval. And hopefully in this calendar year, we'll be able to make the Mighty cohort really the first national prospective trial of what we think will be the most robust marker for early diagnosis of ovarian cancer.
Kevin and Dipanjan, thank you so much for your amazing work and for sharing those results with us. For those of you who have questions that you'd like to ask Dr. Chowdhury and Elias, we are having breakout sessions in just a couple of moments that we are going to transition to. And feel free to join one of those and ask them. We have 4 very exciting sessions. One focused on genetics, which folks may remember was a topic last year. One focused on cancer care during the time of COVID, and another one on cooking to boost your immune system and the importance of nutrition. So I'd like to thank not only our doctors, our sponsors, each of you for coming and joining us tonight, but everyone who has worked so hard to make sure that we can continue our work during this challenging time. If you would like to help us reach our $5,000 match goal, we are getting ever closer. We are now less than 2,000 away. So as Jim and Rebecca said, you can visit tinaswish.org/donate to help us reach that goal. With that, folks should be able to see their breakout sessions. And so feel free to scroll down to those, to click on, and to transition to the next part of our evening. Thank you for coming.